My name's Evan John. I'm from the Centre for Crop and Disease Management based at Curtin University. And my FameLab presentation is called Characterising Plant Disease Regulation for Food Security. Now, in the modern world, we, at least those of us from the economically developed countries, tend to take for granted the food that we eat. Day to day, I certainly do. But if you ask any farmer, they'll tell you that those neatly stacked supermarket shelves hide the real effort that it takes to keep them full. They're also serious threats to our current food supply as it is. As increasing human populations and climate change mean that we need to be even more productive from the limited land that's available to us. But there's also another threat, a pandemic, in fact. But this one's a bit different to the one currently grabbing our attention. Basically, plants, like humans, also get diseases. And by extension, those diseases impact the production of the food that we grow. In fact, it's estimated up to 40% of our food is lost as a result of these pests and diseases. So if we can reduce the impact of these plant diseases, then we'll go a long way towards securing our food production in the future. But how can we achieve this? Well, we've already seen that through the characterisation of human diseases at the molecular level, this provided us with a means to design effective treatments. For example, we have the COVID-19 vaccinations or antiretroviral AIDS treatments, which they've been developed in this way. In a similar manner, the fundamental insights into the nature of plant diseases provide us with an opportunity to protect our food. So I study a pathogen that causes a disease of wheat. It's known as Septoria nidorum blotch, and wheat, being the most widely cultivated food crop in the world, it's used to create things like this, and this, and this, and even this. All right, so the blotch disease I study, it's a problem in most of the regions where wheat is grown. The pathogen responsible is itself actually quite sophisticated, and it's evolved a way to hijack the plant immune system. It does this by producing molecules that cause something comparable to an allergic response in the plant, which then damages the plant and allows the pathogen to take over. So we know that the production of these molecules is tightly controlled to maximize their potency during the infection. But how this is controlled wasn't well understood. However, my work provided some key insights. I was able to characterize the mechanisms of an important regulator, which could be targeted to inhibit the disease. I also characterized the mechanism underpinning how these disease molecules are produced at different rates across the species. So we can now monitor this to help farmers deploy the most effective wheat varieties to be resistant in the regions where it counts the most. So I hope I've convinced you that while serious challenges still remain when it comes to food production, there are still serious opportunities to solve some of these challenges through fundamental scientific research. So thank you for listening.